The scientific method is employed when creating and executing an experiment. Scientists use the scientific method to answer questions and provide explanations about natural phenomena. Following a logical process, we can describe the scientific method in 7 basic steps. To help describe each step, we'll use an example from the hypoxia study with viper shrimp. By the time we've completed this case study series, you should feel confident that you understand how important the scientific method is to maintaining the rigor of discovery and solving problems scientifically. I'll briefly list the steps, and then we'll discuss each one separately with examples. Now, the step on variables is listed as number 5, but actually variables are determined during the experimental design stage. But we'll discuss their importance after observations and before analyses because it's easier to understand them at this stage, and it's important to understanding experimental design. In the first step, an observation provides questions about an observed phenomena. This question might involve an important step that needs to be solved or simply something that will evoke our curiosity. An example of a question would be whether lowered oxygen availability to tissues or hypoxia would have detrimental effects on aquatic animals. Your observations or previous knowledge help suggest this might indeed be the case. The second step is to construct a formal hypothesis, which is a tentative explanation for what was observed. Hypothesis statements should provide a prediction, for example, an if-then statement. In this example hypothesis, we can predict that salamanders develop curved tails if they are exposed to pollutants in the water. And this example given fits the definition of a hypothesis, which is a potential explanation to a question that has been posed. We can develop an if-then situation for this hypothesis. In other words, if pollutants are added to the water, then we would see an rise in curved tails in salamanders. With regards to a hypoxia study, we might predict slowed or stunted growth in aquatic animals, worsening with increasing hypoxic conditions because it is known that all aerobic animals require oxygen to survive. And this hypothesis would then need to be tested to see if observations do indeed support that prediction. The third step is to determine if the hypothesis is valid. And in order to do so, it's necessary to investigate. A defined set of experiments intended to either support or disprove the hypothesis will need to be designed carefully. It's important that the experiment will answer the question unambiguously. For a hypoxia study, an investigator might set up 2 or 3 different levels of oxygen dissolved into water tanks with an indicator aquatic species. An indicator species is known to be highly sensitive to environmental changes. In this case, we'd be looking at fluctuations in oxygen availability. And the African viper shrimp is a good example of an indicator species. Thus, in step 3, we are looking to design an experiment to support or not support the hypothesis. Fourth, an important part of an investigation is to take observations of experimental outcomes. This can be either through sensory perception or through use of scientific equipment that helps detect changes and measures the differences. Aquatic animals might be observed to show distress or possibly stunted growth or other effects in a tank that has been set up under varying hypoxic conditions. In a well-designed experiment, we will allow for observable changes that provide knowledge to help solve the original question. Our 5th step looks at the variables that had been manipulated and shown to have some impact through experimental observation. These variables are factors that provide results to be analyzed in the next step, so we'll go ahead and discuss them here. The independent variable is the factor that is changed by the investigator. It's advisable to use only one independent variable or treatment type at a time. All other factors should be kept constant. In a hypoxia study, the independent variable is identified as dissolved oxygen levels in water tanks. high, normal, low, or extremely low dissolved oxygen levels all represent the same independent variable, just set at different values. As the variable is changed, the investigator will observe what happens. Dependent variable responds to changes made in the independent variable. It is thus dependent on the value set for the independent variable. A good experiment should also include one other factor, which is known as the control, and in which neither the independent variable nor the dependent variable is expected to change. The control treatment confirms that without having made a change to the independent variable, the dependent variable is observed to have not changed. In the sixth step, data collected from the experiment will be analyzed and evaluated according to the original hypothesis statement. To do a statistically relevant analysis, experiments typically include more than one sample or specimen and repeated trials. Larger sample sizes helps to determine whether differences that were observed by the treatments were significantly different and were not just due to random chance or random variation found in nature. The various types of statistical measures will be discussed at a later date. Here we see some data collected from a hypoxia study in coastal waters at different times of the year. Data is analyzed to show distinct times of the year when low DO, or dissolved oxygen, has impacted the aquatic animal populations. From performing the analysis, investigators can make interpretations, and this leads to the final step, a conclusion on whether the original hypothesis was rejected or supported. Generally, when the hypothesis is supported, then the investigations are considered successful. However, even when the hypothesis has been rejected, the investigation may have given important insight into the original problem, which may prove useful to later investigations. Thus, the conclusion asks the question, do results support the original hypothesis given? If the hypothesis was rejected, by default, the null hypothesis is then supported, which basically means there was no change attributed to the manipulations of the independent variables by the investigators. In this graphic interpretation, the investigators' conclusion indicated decreased oxygen levels dissolved in water, resulting in hypoxia, which was due to agricultural runoff into the waterways. Oftentimes, at the conclusion of an experiment, new questions have been posed regarding whether or not a new study should be done to elaborate, to perhaps test out another independent variable, and overall to see what was learned from the experiments. Let's review to see if you remembered all 7 steps by listing them here once more. And so we start off with a question that is posed, leading to a possible explanation or hypothesis, and the opportunity to perform experiments to test those predictions of the hypothesis through observations, having altered or manipulated independent variables, and which provides some data for analysis, and then the analysis leads to interpretation and final conclusions. Now let's take a look again at that null hypothesis once more. When our experiment didn't support the predictions of our hypothesis, by default, we had to accept the null hypothesis. As it turns out, since the null hypothesis is the default, any other explanation are actually considered alternate hypotheses. In the example we have here, we can see a question, why are my socks missing? And possible alternate hypotheses to the null. We can say that aliens stole them, and then provide a prediction through an if-then statement, which would consist of, if aliens entered the house, then my socks will be missing. However, if after an investigation no aliens could be found, and yet the socks still are disappearing, then you would have to reject that alternate hypothesis and accept the null hypothesis, which remains that no change had actually occurred due to those conditions. or more specifically, those predictions. A next step might be to pose a new alternate hypothesis. It turns out investigations remain ongoing and repeated, perhaps by testing a new independent variable, and this repetition supports and strengthens the mounting evidence for any particular hypothesis. New data may always alter or even reject a previously supported scientific explanation. And we always fall back on the null hypothesis when experiments fail to support predictions made. I hope we now understand that the scientific method has a purpose to systematically test ideas and report results in a process of scientific inquiry. A key component of the use of the scientific method is that it ensures that the experiment should be able to be replicated by anyone. If that is not possible, then the results are considered invalid. Thanks for watching. We'll see you in the next video on hypoxia and scientific investigation.